Okay, 2019 paper. Question 4. Cider is made from apples in a process that involves crushing and pressing the apples, converting the sugars into alcohol, maturing and bottling. Brewers add yeast, which contains a mixture of enzymes to convert the sugars in the apples into alcohol and carbon dioxide. State what is meant by the term enzyme. An enzyme is a biological catalyst. That's it. No extra things. Obviously if you're doing biology, you would know a lot more about how it functions. The percentage mass of alcohol in the cider can be calculated using the formula given. A 50 centimetre cubed sample of cider was found to contain 3.05 grams of alcohol. 1 centimetre cubed of the cider weighed 1.36 grams. Calculate the mass of alcohol in the cider. Right, now the only thing that is tricky about this one is the fact that they tell you it's a 50 centimetre cubed sample of the cider, okay, and a 1 centimetre cubed, what that weighs, okay. So the mass of alcohol from the sample is actually 1/50th of that, okay. So for my mass of alcohol, I actually want to do 3.05 divided by 50, and then my mass of cider is fine. Okay, my other option was to multiply up 1 centimetre cubed of the cider by 50, and then my 3.05 would work. Whichever way, as long as you've put in the calculation. So actually what I'm doing is 0.061 divided by 1.36, and that will give me 4. Depending on how far you round, or you could have gone to 4.49, or you could even go as far as 4.5%. Okay, there's your mark. Okay, during the maturing process, malic acid is converted to lactic acid and another product. Name compound X. Okay, so these are the only things that are coming out of it. So let's see what's in the lactic acid, where it came from in the malic. So I've got this bit here is quite obvious, and this is quite obvious, and we're still going great guns. Okay, and then we've got a CH3. So there's CH2, and then I need to take my other H from here, which leaves me with CO2. And there's your answer. Okay, we know it has to be that because there's nothing else going in. That's really what's important to see. The maturing process in cider samples can be monitored using thin layer chromatography. Samples of lactic acid, malic acid and ciders A, B, C and D spotted onto a silica plate, and the solvent is allowed to travel up the plate. Chromatogram is shown below. There we go. Okay, so we've got distance moved by the solvent is 16.4 centimetres, and we've got our point of the solvent is applied. Sorry, not where the solvent is applied, but where a sample is applied. And so it's from this point to here that we're measuring as the solvent front, and then we've got our spots. Okay, and then we've got everything given as stated where they've got to. Retention factor, the RF, is really, it's used an awful lot. This is the kind of standard thing you do with a chromatogram. Because it's a fraction, it means no matter how long the chromatogram is, that fraction would be the same regardless. Okay, calculate the RF value for malic acid. We want the distance moved by the substance divided by the distance moved by the solvent. So for malic acid, okay, we are told that the distance it travelled was 4.1 divided by the distance travelled by the solvent, 16.4, okay, 0.25. And that's it. There's no units because it's basically a fraction. The maturing process is complete when all of the malic acid has been converted to lactic acid. Cider is now ready to be bottled. Use the chromatogram to determine which cider is ready to be bottled. Sorry for the flicking back and forward between. Okay, right, so we are looking for something which has basically got nothing but lactic acid there. So lactic acid is an 8.2 spot. I need something which has none of my malic acid, okay, none of my 4.1. So I am looking for, quite simply, sample 4, cider B. Okay, C, give a systematic name for glycerol. Glycerol can be added to cider before bottling to produce a sweeter tasting cider. It's, you know, they're giving you a new context, but glycerol is just a straight you need to know. That is propan, 1, 2, 3 trial. Nothing else, no other option. Don't try and get fancy and draw it out, because if you draw it out, you've not actually given what they wanted, which is the name. Feel free to draw it out if you've got the picture in your head and you need to then name it, but make sure you've given the name. Okay, cider contains many naturally occurring compounds that affect taste and aroma. You've got procyanidin B2 gives a bitter taste. Explain fully why this chemical is water soluble. Okay, right. For something to be water soluble, then we are looking for something which will basically associate itself with the strongly polar bonds within water. Even better if they also have these bonds, so these highly polar bonds, which will allow, in this case we're looking at that, which will allow your really strong dipoles to exist. And with those really strong dipoles, you can get hydrogen bonds. Well, if you look at this, there is an awful lot of hydroxyl groups. Every single one of those will be polar, and every single one of those will happily make bonds, hydrogen bonds even, not just normal bonds in terms of dipole-dipole, but a very specific one, which will make it soluble. There is a whole pile of stuff in here which does not particularly like it, is non-polar, but the important thing is that it's being outweighed by the water solubility thing. So you want to talk about the polarity of those bonds in there, the hydroxyls, and then how they would associate with water. Okay. Cider smells of apples because it contains ethyl 2-methylbutanoate. Name the carboxylic acid used to make ethyl 2-methylbutanoate. Okay, this is either super easy if you're happy with your esterification, or you can make it a little bit harder but still do the work. Okay, so this bit here, this is originally was your alkanoic acid. Okay, so if I just re-establish that by putting an H in there, that's the acid. And it is 2-methylbutanoic. The ethyl part here was a bit that came from the alcohol. Now, you could just know the fact that you know that. If you know that the ethyl came from the alcohol, then that just tells you it straight away. Or you could name your acid just from the fact you've got the structural formula when you split it in the hydrolysis. Okay, farnesene is a terpene responsible for the ripe apple aroma of cider. Name the molecule in which type terpenes are based. Right, so you're going to get the mark if you say isoprene, just as a straight, that's the units that make it. I think it's, it says name. I think I would be probably better with your 2-methyl buta-1,3-diene. But whichever gets you the mark. What you can't do is just draw it, because if you draw it, you're not naming it. Okay. For E, ethanol in cider can be oxidized to ethanol, spoiling the aroma. Name the functional group circled in the ethanol molecule here. That is the carbonyl. Further oxidation can produce another product that spoils the flavour of cider. Name this product. Okay, so we've got ethanol has been oxidized to ethanol, which is then oxidized to ethanoic acid. And that's your mark. And that's the question.